Hello, today we will be discussing the process of DNA replication. Prokaryotes use the theta mode of replication due to the circular shape of their DNA. This means, as the origin of replication expands into the replication bubble, there will be two replication forks. Looking closer into this replication fork, we can identify the directions of our original DNA strands, which will help us determine in which direction our newly replicated strands will be built. Our bottom strand is 3' to 5', so the top strand, being antiparallel, must be 5' to 3'. First, helicase enters the replication fork, separating the two strands of the double helix. Next, the single-stranded binding proteins attach to the original strands and keep the hydrogen bonds from binding. Next, we can look towards DNA gyrase, which ensures that double-stranded areas outside of the replication fork do not supercoil. Once the fork is stabilized, RNA primase enters, creating primers which tell DNA polymerase where to begin. From there, DNA polymerase III locates the primers and adds nucleotides from the 3' to 5' direction. Due to the continuous replication and direction towards the fork, we can identify the top strand as our leading strand. On our below lagging strand, DNA polymerase still builds in the 3' to 5' direction. However, you can see that this is heading away from the replication fork. Considering this, RNA primase places a path of multiple primers for DNA polymerase III to follow, resulting in discontinuous replication. These pieces of DNA are referred to as Okazaki fragments. Next, DNA polymerase I enters and replaces RNA primers with DNA nucleotides. Along with that, ligase enters and glues the DNA fragments using a phosphodiester bond. Our end product is two DNA double helices, both having one original DNA strand and one new DNA strand, also known as semi-conservative replication.